Advanced Engineering Physics. Unit 1. Crystallography and Materials Characterization. Materials characterization techniques. Until now, we have studied crystal structures, crystal defects, and nanomaterials. However, an important question still remains. How do scientists actually observe the arrangement of atoms inside a crystal? How do they determine crystal structures, measure crystallite sizes, or visualize materials at the nanoscale? To answer these questions, scientists use various materials characterization techniques. These techniques help us investigate the structure, morphology, composition, and properties of materials. Among the most important characterization techniques are X-ray diffraction, commonly called XRD, and Scanning Electron Microscopy, commonly called SEM. Let us first study X-ray diffraction and Bragg's law. The arrangement of atoms inside a crystal cannot be observed using ordinary visible light microscopes. The reason is that visible light has wavelengths ranging from about 4,000 to 7,000 angstroms, which are much larger than the spacing between atoms in a crystal. The distance between neighboring atoms in a crystal is usually of the order of 1 to a few angstroms. To observe such tiny distances, we need radiation whose wavelength is comparable to the interatomic spacing. This requirement is perfectly satisfied by X-rays. X-rays possess wavelengths ranging from approximately 0.1 angstrom to 10 angstroms, which is almost equal to the spacing between atomic planes in crystals. Therefore, when X-rays fall on a crystal, the regularly arranged atoms act like a three-dimensional diffraction grating. As a result, X-rays are diffracted, producing characteristic diffraction patterns. By analyzing these diffraction patterns, scientists can determine the structure of the crystal. This principle forms the basis of X-ray diffraction, or XRD. The phenomenon of X-ray diffraction was explained by the British physicist William Lawrence Bragg. Bragg proposed that diffraction can be understood as the reflection of X-rays from parallel atomic planes inside a crystal. This explanation led to one of the most important equations in crystallography, known as Bragg's law. Let us understand how Bragg's law is derived. Imagine a set of parallel atomic planes inside a crystal. The distance between two adjacent planes is represented by d. Now consider a monochromatic beam of X-rays having wavelength lambda falling on these planes at an angle theta. Part of the beam is reflected from the upper plane. Another part penetrates deeper and is reflected from the plane immediately below. The lower reflected ray travels an extra distance before emerging from the crystal. This extra distance is called the path difference. Using simple geometry, the path difference is found to be equal to 2d sine theta. That is, path difference equals 2d sine theta. For the reflected waves to reinforce each other and produce a strong diffraction beam, they must interfere constructively. Constructive interference occurs only when the path difference is equal to an integral multiple of the wavelength. Therefore, path difference equals n lambda, where n is an integer called the order of diffraction. Substituting the expression for path difference, we obtain n lambda equals 2d sine theta. This equation is known as Bragg's law. It is the fundamental equation of X-ray diffraction. Here, n is the order of diffraction, lambda is the wavelength of the X-rays, d is the spacing between adjacent crystal planes, and theta is the glancing angle, or Bragg angle. Bragg's law tells us the exact condition under which diffraction occurs. If the wavelength and diffraction angle are known, we can calculate the interplanar spacing. Similarly, if the crystal structure is known, we can determine the wavelength or diffraction angle. Thus, Bragg's law serves as a bridge connecting the geometry of the crystal with the diffraction pattern. Now, let us discuss an important experimental technique called the Debye-Scherrer powder method. In the early days of crystallography, diffraction experiments were performed using large single crystals. However, obtaining large defect-free crystals is often difficult. To overcome this problem, scientists developed the powder method. In this technique, the material is converted into a very fine powder and exposed to monochromatic X-rays. The powder contains thousands of tiny crystallites. Each crystallite is randomly oriented with respect to the incident X-ray beam. Because of this random orientation, some crystallites will always satisfy Bragg's law for a particular set of crystal planes. These crystallites diffract the X-rays strongly. Now, consider all the crystallites satisfying the same diffraction condition. Since they are oriented randomly in every direction around the incident beam, the diffracted X-rays do not form a single beam. Instead, they spread out to form the surface of a cone. This cone is called the diffraction cone. The angle subtended by this cone is 4 theta. The powder sample is placed at the center of a cylindrical camera. The inner surface of the camera is covered with photographic film. When the diffraction cones strike the film, they produce circular or curved lines. These lines are called Debye-Scherrer lines. The positions of these lines provide information about the diffraction angle. Suppose S is the distance measured along the film between two diffraction lines produced by the same cone. If R is the radius of the cylindrical camera, then the diffraction angle can be calculated using the relation S divided by 2 pi R equals 4 theta divided by 360 degrees. From this equation, theta can be determined. Once theta is known, Bragg's law can be used to calculate the interplanar spacing d. Therefore, the Debye-Scherrer method provides an efficient way to determine crystal structures using powdered samples. Now, let us discuss how X-ray diffraction can be used to calculate crystallite size. When a material consists of large crystals, the diffraction peaks are sharp and narrow. However, when the crystallite size becomes very small, especially in nanomaterials, the diffraction peaks become broader. This broadening occurs because smaller crystals have fewer atomic planes contributing to diffraction. The broadening of diffraction peaks can be used to estimate the average crystallite size. This is done using the Debye-Scherrer formula. The formula is d equals k lambda divided by beta cosine theta. Here, d represents the average crystallite size. K is called the Scherrer constant, or shape factor. Its value is usually taken as 0.9. Lambda is the wavelength of the incident X-rays. Beta is the full width at half maximum of the diffraction peak. This quantity must be expressed in radians. Theta is the Bragg angle corresponding to the diffraction peak. According to this equation, the crystallite size is inversely proportional to the peak broadening. This means that broader peaks correspond to smaller crystallites. Thus, X-ray diffraction not only reveals crystal structure, but also provides information about crystallite size. This makes XRD an extremely powerful tool for studying nanomaterials. Now let us move to another important characterization technique known as Scanning Electron Microscopy, or SEM. The optical microscope has played a major role in science for centuries. However, its resolution is limited by the wavelength of visible light. Visible light cannot resolve structures smaller than approximately 200 nanometers. To observe much smaller structures, we need radiation with a shorter wavelength. Electrons have wavelengths that are much smaller than visible light. Therefore, electron beams can be used to obtain images with extremely high resolution. This idea led to the development of the Scanning Electron Microscope. SEM is an imaging technique used to study the surface morphology and topography of materials at very high magnifications. It can produce detailed images of structures ranging from micrometers down to a few nanometers. The basic principle of SEM is simple. A highly focused beam of electrons is generated and accelerated towards the sample. The electron beam is then scanned systematically across the surface of the specimen. As the electrons strike the sample, they interact with the atoms near the surface. This interaction causes the emission of various signals. Among these, the most important are secondary electrons and backscattered electrons. Secondary electrons are low-energy electrons ejected from the surface atoms. They provide detailed information about the surface topography. Backscattered electrons are high-energy electrons that are reflected back from the sample. They provide information about the composition and atomic number of the material. These emitted electrons are collected by an electron detector. The intensity of the detected electrons depends on the position of the scanning beam and the local properties of the sample. A computer processes this information and constructs a highly magnified image of the surface. Thus, SEM produces real space images showing the shape, texture, and structure of materials with exceptional detail. Let us now understand the major components of an SEM. The first component is the electron gun. The electron gun generates electrons and accelerates them using high voltages. The electron beam energy may range from a few hundred volts to as high as 100 kilovolts. The second component is the electromagnetic lens system. These lenses act similarly to optical lenses. They focus the electron beam into a very fine probe, having an extremely small diameter. The third component is the scanning coil. The scanning coils deflect the focused electron beam in a raster pattern, causing it to move systematically across the sample surface. The fourth component is the specimen chamber. This chamber contains the sample that is being analyzed. The fifth component is the electron detector. The detector collects the secondary electrons and backscattered electrons emitted by the sample. Finally, the detector signals are processed and displayed on a monitor as a high-resolution image. SEM offers several advantages. It provides very high resolution. It has a large depth of field, allowing three-dimensional surface features to be observed clearly. It can examine a wide variety of materials including metals, ceramics, polymers, semiconductors, and biological specimens. SEM is widely used in materials science, nanotechnology, metallurgy, electronics, forensic science, and biomedical research. To summarize, materials characterization techniques are essential for understanding the structure and properties of materials. X-ray diffraction uses the diffraction of X-rays by atomic planes to determine crystal structure and interplanar spacing. Bragg's law, given by n lambda equals 2d sine theta, provides the condition for constructive interference. The Debye-Scherrer powder method enables diffraction studies using powdered samples. Peak broadening in XRD can be used to estimate crystallite size using the Debye-Scherrer equation. Scanning electron microscopy uses a focused electron beam to produce highly magnified images of material surfaces. Together, XRD and SEM form two of the most important tools for investigating materials at the atomic, microscopic, and nanoscopic scales.